Good morning and hello everyone. I'm Professor Sonali Pawar from JSP College of Pharmacy and Research, Bakuli. Today I deliver a lecture on a polarimetry. So here we are in a pharmaceutical analysis 2. So what exactly polarimetry is, we learn in this topic. So here we are, and we are introducing first what is exactly polarimetry, then what is the term plane polarized light, then what are the optical activity, then third theory of optical activity, polarimeter and applications. So now number one is introduction part. Polarimetry is one of the important instrumental method. This is the instrumental method and in the analysis. This measures the rotation of the polarized light. Very important part is that what exactly it measures, that rotation of the polarized light as it passes through an optically active compound. Whenever we are seeing what I said, this optically active compound or not, so we are calculating from this method or this instrument. This technique involves the measurement of a change in the direction of a vibration of a polarized light when interact with an optically active compound. A substance is said to be optically active if it rotates the plane of the polarized light. If you see here this light, when we are switching the light, what exactly the ordinary light is come and vibrating a various different direction, and in a polarizer we are this light passes as a plane of a polarized light. In presence of optically active compound, we are see the levorotatory compound, that means it denoted with a negative part, or a dextrorotatory part, that is a positive point. So next second part, the plane polarized light. So what is the exactly plane polarized light is it? According to a wave theory of the light, an ordinary ray light is considered to be a vibrating in a all plane at a right angle to the direction of a propagation. If this ordinary ray of a light is passed through a nickel prism, which where is located in this instrument, the emergent ray has its vibration only in a one plane. This light having a wave motion in a only one plane is known as plane polarized light. Nickel prism is mainly manufactured for many more things, like Iceland spar, chalcolite and polarite. A plane polarized light which consists of two components of a fixed magnitude rotating in opposite directions to one another. The right circulating polarized light and second part is the left circulating polarized light. Plane polarized light is the vector sum of these two components, if you see here in the diagram. So, the term polarimetry may be defined as the study of rotation of plane polarized light by transparent substance, and ordinary light usually vibrates in all planes. So when we are putting the sample in the instrument, what exactly happened? The unpolarized light is come from the light source, then with the help of nickel prism it goes in a plane polarized light, and then after we are calculating it is rotating the substance, that means we are calculating the optically active compound and we are calculating the optically inactive compound with this help of the linearly polarized light. So what is exactly polarization? It is polarization is nothing but one type of the electromagnetic waves are vectors, have an intensity and a direction of a propagation associated with them. Here we are, just see this diagram. What exactly it noted that transfer to this plane is the plane in which the electrical and the magnetic field oscillates. Imagine one monochromatic then infinite harmonic wave propagation in some direction. If the direction of a E field is unchanged, that is, it is always perpendicular to the direction of propagation, but let's say it always point up as well. The wave is linearly polarized. Here, see this diagram. What exactly happened here? Polarized filter, that means the nickel prism is there. what exactly happened? The many more unpolarized waves that come after filter, the polarized waves is passed out. Linearly and the circularly polarized radiation are specified cases of a elliptically polarized rotation. In this case, the tip of the elliptical vector traces out an ellipse viewed at one place in space. The general case of this arbitrary phase E, here seen in this diagram, between our two orthogonal waves at arbitrary amplitudes gives us elliptically polarized radiation. So this is the elliptical polarized radiation. The locus on the tip of this elliptical vector is referred to the polarization ellipse. So we now know how to refer to this polarized state of the monochromatic wave, and that the general case of elliptically polarized radiation can be decomposed into two orthogonal, unequal amplitude, linear, or the inner circular states. So next point is, what exactly optically activity? So here the definition is, it has find that magnitude of a rotation depend upon the following factors. Number one, nature of substance. What exactly the nature of this substance? Number two, length of liquid column through which light is passes, which were located in your instrument. Concentration of this solution. What exactly concentration you are used at the time of the practical purpose. Nature of the solvent, and then temperature of the solvent and wavelength of the light you are used there. Types of molecules analyzed by polarized molecule must be optically active, and only the optically active molecules contain the asymmetrical carbon atoms. The rotation power of a given solution is generally expressed as a specific rotation. This is a very important term, specific rotation. In a many time in exam purpose, this question is asked, what exactly the specific rotation is it? It is the number of a degree of a rotation of a plane polarized rise produced by 1 gram of the substance per ml, which were in the solution. The measurement is carried out at a temperature using sodium light. This sodium light is very important for this practical purpose. The specific rotation can be calculated by the following relation. So here is the formula. Then specific rotation, temperature, wavelength, optically rotation, concentration, and optically path length in the TM. In just before slide, we learn about it, what exactly we need that, and here is the formula for that. 100 multiplied with the observed angle of rotation divided by length in the decimeters multiplied with a gram of a substance in 100 ml of the solution. So next is the theory of the optical activity compound. Everyone know that in a chemistry this is a very important part, that's the extrorotatory and the levorotatory. An optically active substance is a one that rotate the plane polarized light. Here is the example: lactic acid, dextrolactic acid, and levolactic acid. What exactly here? One type of the mirror image kind of the structure we are seeing here. So if it rotate at a right, that means it's a, or the clockwise direction is a dextro. If it rotate at a left side or a counterclockwise, that means it's a levo. That means levorotatory compound, and we are observing if this kind of two compound are there, that means optically active compound it is. So what exactly instrumentation is here? It is a very important point. So optical activity may be detected and measured by an instrument, and this instrument is called as polarimeter. Light source is a sodium vapor lamp. Analyzer, another nickel prism aligned to the intercept the linearly polarized ray as it emerges from the sample solution. This is the work from the analyzer. That means in this instrument we know the light source which we are using, analyzer, what analyzer we are using, and number third, a polarimeter consists of a polarized light source, sodium vapor lamp, polarizer, quartz prism material, and analyzer is nickel prism aligned to the intercept the linearly polarized rays as it emerges from the sample solution. A graduated circle to measure the rotation angle and sample tubes. Here is the polarimeter. From this object we are seeing the with the help of our eye view what exactly degree we calculated. After the practical performances, we are seeing the text to and the logo, and we are calculated with this help of the specific rotation program. So those are the different parts what exactly we are using in instrument: analyzer, polarimetry tube, fixed polarizer, light sources. Here is the polarimeter, how it look like, and this is the one type of this part: eye view, that means the viewer; analyzer, sample tube, where is the sample is located; then polarizer, then the light sources. Optical rotation power. This is also known as the ORT, useful for the qualitative analysis, quantitative analysis, elucidation of chemical structures. Unpolarized, the vibrations may all be restricted to the one direction only in the particular plane. What is the plane polarized light? Is unpolarized light get converted into the plane polarized light by simply passing it through a lens called as a nickel prism. After William Nicol, this is an inventor. Nicol prism is a isolate sperm, calcite, polaride. A plane polarized, which consists of the two components of fixed magnitude rotation in opposite direction to one another. The rotation circular polarized light and the left polarized light. The plane polarized light is a vector sum of these two components. So this is the one type of the summary of this part. What are the applications for this practical? Quantitative analysis is happened, qualitative analysis is possible, and saccharimetry is possible. What is the exactly saccharimetry it is? Saccharimetry is an important practical application of a polarimetry, determination of a high concentration of a sugar. Various concentration of a sugar is calculated with the help of this practical and by using this instrument. Visual saccharimeter is called as polariscopes. Polarimetry industrial application. Research application for a polarimetry. Many more industrial research institutes are using this instrument for this part. Many high HPLC with the help of HPLC and many more this polarimetry are used for various purposes. Using polarimetry in a quality and a process control application too. Control application, both the laboratory or an online in the factory, are found through the pharmaceutical, essential oil, flavor, food, and chemical industries. In many more time it's used. So, pharmaceutical industries, what exactly we are determining with the help of this part, with the help of the specific rotation practical performance and the optical rotation practical performance. Amino acid, antibiotics, dextrose, steroids, amino sugars, cocaine, diuretics, tranquilizers, analgesics, codeine, serum, vitamins are different products we are calculating with the help of this instrument. Polarimetry for flavor, fragrances, and essential oil industries also use this for this determining the camphor, gum, orange oil, citric acid, lavender oil, spearmint oil, glycolic acid, and lemon oil too. In a polarimetry for the food industry's application, polarimetry ensures this product quality by measuring the concentration and purity of the following compound in sugar-based food, cereals, and the syrups: carbohydrate, lactose, raffinose, various starches, fructose, levulose, sucrose, natural monosaccharides, glucose, maltose, xylose, many more. Using this polarimetry in the chemical industries too, polarimetry analyzes optical rotation as a means of identifying and characterize the biopolymers, natural polymers, and synthetic polymer too. Thank you so much to listening this whole chapter, this polarimeter. What are the exactly it is in a depth we are learning here. Thank you so much for this. Thank you very much.